So Dr. Suzanne Drury is next from Congenica. We've heard about Congenica a few times today. Clinical decision support for genomics at scale. Thank you.
Good afternoon, everybody. I'm really pleased to be here. Thanks ever so much for the invitation to present. And it sort of occurred to me as I was putting these slides together that about 15 years ago, I was working in an academic post, and I was looking at metabolizer status of CYP2D6 and whether there was any association with recurrence of breast cancer. And then about 8 years ago, I was working in the then regional genetics labs, and this was at a time when next-generation sequencing and the sort of higher throughput genotyping assays were becoming available in the clinic. And at that time, we were starting to explore whether or not there was any appetite for the implementation of pharmacogenomics in the tertiary setting. And at that time, there wasn't any appetite. So now here we are, 2022. I think it's the fact that we're all in this room together having this conversation really shows that the time is now for pharmacogenomics in clinical care. So I'm going to go through, I guess, a little bit of a journey, really, about Congenica and kind of explain how our experience over the last few years, I think, now puts us in a really good position to be able to help deliver a solution for the implementation of pharmacogenomics. So Congenica, we harness the power of genetic data to drive precision medicine worldwide. And that comes in many flavors, really. So we are focused on rare disease and how rare disease diagnosis can ultimately inform therapy. We are focused on oncology, and now we're going to be focused on pharmacogenomics as well. Just to give you a bit of a background, Congenica is a company which was born out of work at the Wellcome Trust Sanger Institute, along with colleagues in the NHS. And it was really sort of evolved from the Deciphering Developmental Disorders study several years ago. And that study was really looking at the implementation of new genomic technologies, at the time array, which subsequently turned into exome sequencing, but how those new technologies could improve diagnosis and patient care. And the founders of Congenica basically realized that there was going to be a need for a platform to be able to manage these increasing amounts of genetic data and to be able to turn that into basically a product which could be implemented in clinical care rather than just in the research space. So Congenica was founded in 2012, and in 2015 the rare disease product was launched. And we're a key partner in the Genomics England and the 100,000 Genome Project. And we were involved all the way through from the pilot phase to the actual rollout of genomic sequencing in the NHS, to the point that in 2018 we secured the exclusive NHS tender. And so basically all of the whole genome sequencing that is carried out in the UK, all of those results come back through the Congenica platform. In 2021, the product achieved the CE mark. So we heard about some of the regulatory requirements earlier on today. So we've been through that process with our rare disease and also our oncology platforms. 2022, so this year, along with various other successes, we've been awarded a role in the Hong Kong Genome Institute whole genome sequencing projects. We have our somatic oncology launch a little bit later on this year. And also we've been awarded a phase 1 tender with the UK HSA, so that is part of the pathogen sequencing, so as a result of SARS-CoV-2 sequencing. So we're really sort of expanding out from that sort of those rare disease origins, but an awful lot of what we've learned in that process is now really enabling us to go into these other areas at scale. So it's just, and I'll touch upon these a little bit more a bit later on, but some of those key features, which I think are also going to be really applicable to the rollout of pharmacogenomics, is automation. So going from your test, your assay result to a report, whatever that report might look like. That could be a report that's a simple piece of text, or it could be a full-scale report. being able to sort of generate those results in a way that is able to integrate with EMR or EHR systems. Scalability has been incredibly important, to be able to process, thousands of samples a week. And, I think we can see that even if that's not the starting point for pharmacogenomic implementation, it will very quickly become necessary to be able to scale. And also thinking about kind of embedding pharmacogenomic results with other genetic testing that is going on as well. So you saw this slide earlier. Polly from First Data Bank presented this slide. So just looking at the sort of the higher level pathway where we might see a patient arriving for a medical review, where a test request is prompted, which, as Polly indicated, could happen within the FDB system. The lab process happening. So I think obviously at the moment we're not completely sure, I think, within the UK as to exactly what that looks like, but it's probably going to be a panel of some sort, whether that's a variant panel or a gene panel. And likewise, what the actual assay itself is going to look like. But basically there's a lab process, and then you need to be able to convert that raw data into something meaningful. So there's this box of analysis, and I shall show you a little bit of the detail of that in a second. You get a result, and then that needs to feed back into the EHR system and the e-prescription. And then, of course, you have your medical review, and then basically we need to have the ability to re-query that data in the future. So this is sort of some of the steps that need to happen to translate that genomic data into treatment guidance. The data has to go from raw data into genotype, into a haplotype, into a diplotype, into a phenotype, and then into guidance. Now, this is just some of the experience, I guess, that we have from Congenica, which we think is going to be applicable to be able to help this pathway along. So the data that's generated, obviously it needs to be stored, and it's probably going to need to be stored for a very long time to make it requeryable. Obviously, it's going to be genomic data, so it needs to be stored securely, and it really needs to be scalable. So it may be, obviously initially it's probably going to be a few variants for a few people, but that's going to mushroom over time. And that certainly presents an engineering challenge, and that's an engineering challenge that Congenica have already overcome by having to deal with 70,000 human genomes, which is a huge amount of data. And sort of importantly as well, from a data storage point of view, you need to be able to store it in such a way that it can be accessed, and it can be accessed quickly. You don't want these processes to kind of be slow when you're requerying information. We have systems which can be stored locally, or we also have a system on the cloud, and that's stored in the UK. And in terms of the actual kind of beyond the storage and thinking about data itself, then I've mentioned it needs to be requeryable. We've already heard about coding, and whether that's SNOMED coding or whether that is any other coding that guidance recommends. And obviously maintaining reference data. And we have a lot of experience with maintaining reference data and keeping that up to date and auditable, managing releases and so on. So we have over 40 different reference data sets in the rare disease platform, just as an example. We are very experienced in terms of that QMS process that you heard earlier on from the MHRA presentation. We have a completely embedded QMS system within the company, so the software that we develop is regulated according to all the best practices, including NHS. So just to sort of reiterate that at this moment in time, then it's the rare disease and the oncology platforms that do have that CE marking, but obviously the development of the pharmacogenomic platform will have to adhere to those regulations, which, as was pointed out earlier, is in a sort of state of flux as well. And finally, just to say on this slide that we have a lot of experience around implementing AI and machine learning as well. So we have that skill set to bring. And also we have a lot of experience of sort of evolving with technology in terms of the different laboratory assays that are used. So this slide just kind of shows you really, I guess, kind of the availability of genetic data in the different care settings at this moment in time. So obviously at the moment, most genetic data really sort of sits within in the tertiary care, that kind of rare disease space where we're seeing those whole genomes. Secondary care, obviously oncology, we're starting to see the implementation of genomics increasingly in that space. But obviously the big opportunity is really in primary care when it comes to the number of patients who might benefit from this testing. A study from the UK Biobank showed that essentially all of us will carry at least one variant which would predict a non-typical drug response, and actually the average atypical dose guidance was for about 10 drugs per individual in that, so it's hugely applicable to all of us. But I just wanted to make the case as well that actually even within sort of the oncology space and rare disease, that the application of pharmacogenomics to, if you like, the slightly more generic drugs are applicable in those spaces as well. So at least a third of oncology patients will be on multiple other medications to manage their symptoms, and likewise rare disease patients. I think a study by the Mayo Clinic, which looked at their predominantly neurodevelopmental disorder patients, where they had whole exome or whole genome sequencing available, they found that about 20% of those patients, again, also on a number of other drugs, essentially on polypharmacy to manage their other kind of symptoms. So I'm not going to spend very long on this slide, but really just to sort of point out, as we've already heard, sort of I guess there's really sort of the inefficiencies in the system, spending money on medications which may not be the most ideal for that patient. So this figure just shows some of the data from the USEF paper, which we heard mentioned earlier, where they sort of projected the number of individuals who would be expected to have a recommendation for an alternative medication. And then some of the costings here, which were generated by data from within First Data Bank. And also we sort of looked at the fact that about 500,000 individuals for these drugs in pink, which you can't quite read, but sort of clopidogrel, simvastatin, venlafaxine, about 500,000 individuals would be recommended to have an alternative medication. And altogether, in that paper, which was looking at primary care, then about £40 million worth of medications could perhaps be redirected should that patient have a pharmacogenomic test available. But some of the other sort of the features, I guess, of a PGX pathway and clinical decision support system that would ultimately, I guess, add value to this testing. One is being able to support panel testing, be that single variant, be that multiple genes, making sure that those results are embedded in some format into the EHR so that they are then there and they are requeryable. So you're not going to have to repeat the laboratory test. I think that's really key. So what are Congenica doing in the pharmacogenomic space at the moment? Just over 18 months ago, we were awarded a grant by Innovate UK, which is the government's innovation kind of funding. And this funding was in conjunction with Legal and General. And that funding is for the development of a pharmacogenomics platform, in addition to a clinical study. So I'll just give you a bit of an outline of this project, which we call HAPPY, which is healthy aging and pharmacogenomics and polypharmacy. And it's the clinical study is aiming to recruit 500 patients who are over 50 years and on 3 or more medications. That recruitment is happening in 2 centres, and I see our recruiters here in the audience today. Thank you ever so much, doing an amazing job, and recruitment is going really, really well. And within that study, we're capturing information on sort of the number of times a patient has been to see the GP or had an A&E admission in the previous year. We're then going to be looking at that figure sort of 1 year post pharmacogenomic testing. We're also capturing patient-reported adverse reactions. And essentially, we're sort of using that study really to help inform what a clinical decision support platform should look like, and really as well understanding those decisions around, if you have a pharmacogenomic result that suggests a change should happen, can it be actioned, and if it can't be actioned, why not? Coming back to, I think, someone's point earlier about, maybe there's not an alternative medication, so we're capturing all of that information as well. And at the moment, we're capturing all of that information in a sort of a standalone patient registry. But obviously, as and when this gets fully embedded into the EHR, all of that sort of information will become queryable. The kind of the PGX platform itself, we fully intended it to be assay agnostic. And we are looking at different secondary and tertiary analysis pipelines to support those different assay starting points. And obviously, ultimately looking to integrate that result to enable timely and situation-specific results. So if we just think about this as the right drug, right dose, right time. So really, what we are developing is an automated workflow from raw data to report. And that needs to be a fully coded pipeline with coded results, for example, SNOMED, or as we said, potentially other codes as well. We want this to be sort of embedded within existing genomic testing services, so thinking about also being able to support analysis with things like genome sequencing as well as an individual panel test. We really believe that this solution needs to be future-proof and dynamic to be able to support the sort of fairly continuous updates in terms of reference data, and also to be able to support the changes in genomic technologies that are likely to be coming through in future years. Because it's one thing to sort of be able to deal with a small number of variants now, and it's another to be able to sort of scale up and be able to consume vast amounts of genomic data. So we need to kind of make sure that we build that solution with that in mind. And we're also conscious of the fact that we need to be able to incorporate other factors like age, as we've just heard, and comorbidity. But I think that's really going to come when we can fully embed these results into the existing health system. Something else we're really conscious of is, we really need to be able to incorporate guidance that's recommended by the NHS. So whilst we could directly take the CPIC or the DPWG guidelines and implement those, we really want to work with the NHS stakeholders to understand what your requirements are and how results should be displayed. And ultimately, again, as we've heard throughout the day, the need for the results to be easy to interpret, and again, that's going to come from stakeholder engagement, and of course, really to be situation-specific at the point of guidance. So you probably would have seen the new 5-year programme that was sort of launched by the NHS maybe 2 or 3 weeks ago, and there's a definite intent within that for pharmacogenomics to be rolled out over the next 1 to 3 years. And Congenica, we have a vast experience in providing fully integrated and automated workflows with the NHS, which we sort of subsequently adopted for various other projects. And as you heard earlier, we started this session with First Data Bank, and now we're ending this session, by sort of really saying that First Data Bank and Congenica will be able to provide a solution for implementing PGX to personalised patient care. And we think the right drug, right dose, right time is really about demonstrating that we will have reliable, recurable, and real-time data. Thank you.
Thank you very much. Questions? I know we're waiting for coffee and so on. John.
Thanks very much. So I know you said that the platform is agnostic of assay. Obviously, we're mostly using SNP genotyping assays at the moment. Obviously, you guys are the experts in kind of looking at sequence data. Have you done much work looking at extracting pharmacogenetic data from sequence data?
That is part of the Innovate UK grant at the moment. Yeah, so we're actively looking at exomes, genomes, panels, and I guess just to say as well from a genotyping point of view, then we are experienced at taking anything from a VCF with a few variants all the way up to whole genome. And okay, that's currently not in the PGX space, but we're completely scalable in that sense. Thanks.
And how are you dealing with 2D6 from short reads?
We're looking at lots of different options at the moment. You probably know there's many different sort of tools out there, and again, that's a specific.
You haven't identified the right one yet.
No, that's a specific piece of work that's ongoing at the moment, and we're sort of looking at various, Coriell samples and so on. And
have you got a question?
As a follow-up question, so your input is always a VCF file, or you can take it all the way down, to a BAM file? Going back to SIP2D6, so you won't necessarily do the haplotyping all the way down to star allele calls, and you're integrating with other technologies?
Obviously, that is very much dependent on the assay itself, but we're also very conscious of the fact that the assay obviously is driven by the laboratory, and if the laboratory assay doesn't include the specified variant set, and again, that kind of variant content is something we really want to work with the stakeholders on, because there's obviously different labs have different minimal content to be able to call those star alleles.
Great, thank you very much. Sorry, Carl, last question.
I have a question about your integration to the EMRs. Is it one of storing the report within the EMR, or are you putting over the genetic information that can be queried directly from the EMR?
So we would be able to provide whatever the EMR actually requests. So it could be just a subset of data, or it could be the whole data set. So very much depends on the actual requirements of the EHR.
Okay, thanks very much.